Good afternoon to all. Myself, Arvind Kumar, Assistant Professor in the Department of Pharmaceutical Chemistry in the School of Maharaja Aghasin School of Pharmacy, Maharaja Aghasin University, Baddi. Today, I am going to deliver a lecture on the molecular modeling virtual screening techniques, that is molecular docking. So, under this, we are going to discuss a drug discovery. What is drug discovery? It will take years and decades for the dissolving or for discovering a new drug, and these are very costly in nature. To cut down the research timeline and cost by reducing the wet lab experiment by using computer modeling softwares like Gold, Schrodinger, etc. Drugs interact with their receptors in the highly specific and the complementary manner. These drugs are in the core of their target-based, structure-based drug design in the lead generation and optimization. So, we are going to discuss what is docking. Docking is an attempt to find out the best match between the two molecules. First is known as your ligand, and second one is your receptor. Docking is a structure-based technique which is attempt to find the best match between the two molecules. As I earlier told that one molecule is your ligand molecule, that is your drug molecule, discovered drug molecule, and the second one is your receptor, which comes in your protein binding. And the docking, the best match, that is induced fit, that they will take a best match molecules. Then we are going to a more serious definition. Docking is a method which predicts the preferred orientation of the one molecule to a second when bound to form a stable complex with the overall minimum energy. Docking. What is docking? Docking is base, and what are the applications? Docking is used to predict the both strength and types of signal produced during the interaction of which drug and the molecule or its receptor. What is molecular docking in the field of molecular modeling? If we are going to in the field of molecular modeling, docking is a method which predicts the preferred orientation of one molecule to the second when bound to each other and to form a stable complex. Knowledge of the preferred orientation in turn may be used to predict the strength of association of the binding affinity between the two molecules using, for example, scoring functions. So in here we are going to discuss there is a receptor and there is a ligand. When the ligand is bind with the receptor, or the receptor is going to bind it with the ligand, there is a formation of the complex, that is receptor ligand complex, and that will form a drug action. Then the drug will act on your body. Then it is a computational procedure to find out the active ligand site. It is also a target based drug discovery. In this, what we will do, it is a computational procedure, okay, in which we will find out the active ligand, which comes under the sitemap method of the docking. Then the molecular docking studies are useful to find out the lead compound which will fit into the receptor effectively. Then the docking studies give reliable information about the perfect orientation of the ligand with the protein and to use same in the rational drug design. What is the molecular? Why the molecular docking is too much important? It reduces the signal transduction. In the signal transduction, the association between the biological relevant molecules such as protein, nucleic acid, carbohydrates, and lipids, which play a central role in the signal transduction. Furthermore, the relative orientation of the two interacting partners may affect the type of signals produced. That example, agonism and versus the antagonism. Therefore, the docking is important for the predicting both the signals and the type of signal produced, what kind of signals are produced and what kind of signals length or band is obtained from by the signal. Here we seen in this slide the adrenaline receptor. The adrenaline drug is bind with its receptor, that is known as adrenaline receptor, and we will find out the GTP is converted into the GTP, that is the diphosphate is converted into a triphosphate, and then the adenine cyclase enzyme will act and the cyclic AMP is produced. In the next, the docking is used in the drug design. Docking is frequently used in the predicting the binding orientation of the small molecules drug candidates and to their protein targets in order to in turn predict the affinity and the activity of small molecules. Hence, docking plays an important role in the rational drug design. Various kinds of drugs and various molecules are discovered with the help of docking, that is comes under computer aided drug design chapters. However, since the both ligand and the protein are flexible in nature, in a hand in glove, all of you know what are the hand the gloves are movable, and it will act as a protein or a receptors, and your hand is act as a any molecule, any ligand, and any, or you will say that any drug molecule. Like also it will appreciate okay, analogically is more appropriate than the lock and key hypothesis. In the lock and key, the particular key is formed for the particular lock, then same as the ligand is particularly for the receptor of the protein. During the course of the docking process, the ligand and the protein adjust their conformation to achieve an overall best fit, and the kind of conformation adjustment resulting in the overall binding is referred to as an induced fit. Now we're going to what are the types of docking. The types of docking are the first is the lock and key or rigid docking. In the rigid docking, both internal geometry of the receptor and the ligand is kept fixed in a docking is preferred or performed. In the lock and key hypothesis or the rigid docking, the both molecules, the receptor molecule as well as the ligand molecule, will fix in their in the space. Induced fit, second one is induced fit, or also known as flexible docking. As the name suggests, an enormous on the rotation of the one molecule, usually smaller one, is performed. Every rotation, the surface cell occupies or occupancy that energy is calculated later than the most optimal pose is selected. In this, both the receptor as well as ligand are flexible in nature. Now coming to the rigid docking, as the diagram or as the figure shows that these are historically the first approaches. Protein and ligand are fixed. They are going to search for the relatively orientation of the two molecules with the lowest energy, that is the protein docking. Both molecules usually considering rigid, that is the your receptor one, and your ligand, that is your drug, the discovered drug. The first applies steric constraints to the limit, search spaces and examine the energies of the possible binding conformations. Now coming to the flexible docking, as the name suggests, there is a coming under the protein ligand docking, flexible ligands and rigid receptors. In these, the receptors and the ligands, receptors which are comes from the protein, as the diagram shows. In the flexible docking, both the protein and ligands are flexible nature. They will search a space much larger, either reduce the flexible ligand to rigid the fragment connected by or several hinges, or searches by the conformation space by using the Monte Carlo method or the molecular dynamics. In these methods, they will automatically adjust according to their flexibility or their receptor size. Now in the flexible docking, the receptors are the ligand and kept flexible, and their associated energy is calculated. This method is good to study about the binding patterns of the drug with the receptors. Now coming to the mechanics, what are the mechanics of docking? To perform the docking screen, the first requirement is a structure of a protein of interest. Usually the structure has been determined by using the biophysical technique such as hybridization or organization, but it is also derived from the homological modeling construction. The protein structures are databasely potential ligands are served as the important docking programs. The sources of the docking programs depend upon the two components, search algorithm and what are the screening functions. In the search algorithm, it is a mathematically model. These are the mathematical models which are generally used to determine the possible orientation of a drug receptor complex and interaction energy associated with it. in the scoring functions, this is the function, or these are the functions which are used to predict the strength of the binding affinity between the protein and protein ligands, etc. In the scoring function, we're going to find out the best pose or the best ligand site by the scoring, or protein interaction by the protein ligand interactions. These configurations of the binding affinity are evaluated by the scoring functions. Now we are going to a typical overview of what is the how the docking will workflow. What is the workflow of the docking? In firstly, we'll target the selection. What is the first step? Will there are two first list first there in these studies we have two subjects, or two mainly targets. First, target selection, and second one is the ligand selection. Under the target selection, the target selection is prepared, or the target is prepared by the docking procedures, and in the various docking procedures, in the various softwares like Gold, Schrodinger, the target is selected. In this, the protein will come and we'll find out the receptor in that protein. Under the ligand selection, we'll find out the ligands, what are the drug molecules that are going to, new drug molecules are situated or formulated. Then after that we will prepare the ligand. That will the first task we will do, the prepare the target and the prepare the receptor site, and then second one is will prepare the ligand. Then both the target and the ligand will attach and the dock. In the simple language we will say that will dock, and then we'll evaluate the docking results with the help of various scores, docking scores, glide score, and their docking poses. Now, what are the applications of molecular docking? With the help of docking molecules, or with the help of molecular docking, we will determine the lowest free energy structures of the receptor ligand complex, and we will also calculate the different binding of the ligand to the two different macromolecular receptors. We will also use, in the molecular docking is also used to study the geometry of the particular complex, and for the purpose of the modification of the molecular or lead molecules of the optimized potency or the other properties, like, and it is also used for the de novo design for the lead generation, various kinds of drugs designing, and will in the simple language there is a library of drugs in the screening for the side effects that can be caused by the interaction with the other molecules, or to check the specificity of the potential drugs against the homologous protein through the docking. Docking is also widely used tool. It is a tool which is very widely used for predicting the protein-protein interactions, and knowledge of the molecular association aid with the understanding the variety of the pathways taking place in the living and in the releving of the possible pharmacological targets. Now, what are some common used docking softwares purpose? What are these docking software? As the earlier said, there's examples of the docking softwares. First, USCF dock, prepared by the USA in the 1988. Second one is Autodoc. It is also prepared by the USA in the 1990. Autodoc is a suit in which automated docking tool. It is predict how small molecules such as substrate and the drug candidates, and they are going to bind with a receptor of a known 3D structure. In this, current distribution of Autodoc consists of two generations of softwares, that is Autodoc Wing and Autodoc 4. Next one is the Flex, made by the Germany in the 1996. In this, the receptor is treated as rigid. It comes under the rigid docking. In this, receptor is treated as rigid and incremental constructions algorithm. In this, break ligand up into the rigid fragments, dock fragments into the pocket of the receptor, assemble ligands from the fragments. It is also very useful for the energy conformations. Next one is the dock, that is next docking software is used as Gold, naming as Gold, that is made by the UK in 1995, that will perform automatic docking with the full acyclic ligand flexibility, partial cyclic ligand flexibility, and the partial protein flexibility in and around the active site. Other softwares are used ADDS made by the India in 2011, ADMS made by Japan in the 1994, Betadoc in the South Korea, Darwin in the USA and Diwali by the USA. Doc Vision is made by the Canada. Now we are going to what are the different approaches. There are 2 to 3 approaches. First, we will discuss the receptor or the structure based approach. In this, we'll find out the active site identification. Firstly, we are going to find out the active site identification, where are ligand going to attach or will induced with their site. So in the first step, we'll find out the active site identification, then ligand fragments group, then we'll going to the ligand fragment grouping. Under the ligand fragment grouping, the ligands are fragmented and then grouped into the different kinds of receptors, which will going to bind with the receptor, and then we'll check the ligand with attached to the receptor. They will, in the simple language, will find out the ligands or our drugs will going to attach with their active sites, that is comes in the receptor or not. They'll fit to the receptor or not. If they will fit to the receptor, then we'll go to the complex growing. We'll find out how the complex is grow under the receptor, and then it will go or give the positive results, then the potential drug, or we can say that the drug comes after that in the market, or then the drug is going to made for the synthesize, and then for the various market studies, that is undergoing to the clinical studies and not. If the complex growing is not given to the success, is not going to the successfuls, then there is a problem in the ligands. Active site is okay, but there is a problem in the ligands. Then the complex will go. Then in the complex growing, if they will go the negative results, then we will find out what is the problem in the ligand. Then we will verify or clarify the ligands. Then there are changes in the ligand. Then with the various kinds of groups, we'll change the groups, and then after that we will fit in the receptor. If they will not fit in the receptor, then we'll change the receptor. Then next is under the ligand based approaches. Under the ligand based approaches, there is the pharmacophore identification. After the pharmacophore identification, means after the selection of the pharmacophore, we will modify the pharmacophore. In the modification of the pharmacophore, we will give the structures and the receptors. When the structure and the receptors will fit in, when the ligand is going to fit in the receptor, then the potential drug will come out. If there is if the ligands do not fit into the receptors, then they will back return the pharmacophore modeling or pharmacophore modification. What is docking? What are some important steps in the docking? First, ligand preparation. As we already told, ligand preparation. What are the ligands? Ligands are your drug molecules. Ligands are your drug molecules which are discovered, which are going to be discovered. Next, and under this category, we will prepare the ligand and by using their softwares. Next is the protein preparation wizard. Under the protein preparation wizard, we will prepare the protein. In the next slide, in the coming slides, I will explain it. Next, site map, receptor grid generation, docking, evaluating with the docking score. In this docking score, and what are the poses that are very important. Now we are going for the firstly ligand preparation. In the ligand preparation, this kind of ligand preparation wizard comes. Okay. In the ligand preparation, firstly we will prepare the ligands. In this, we will prepare the structure with the help of various softwares like ChemDraw, PubMed. Okay. In the ChemDraw, you have the ChemDraw. The ChemDraw is a software which is used to prepare the structures. By using the ChemDraw softwares, we will make the structure, and we will going to prepare our ligand. In the ligand preparation, it is simply the ligand we will prepare is going, when the ligand is going to prepare by the software language. In the software language, what is the software language? In this, we will find out the ligand is going to the attach, what are the changes and what are the poses given to the receptors. And what is the receptor or protein will take the poses from the ligand, they will attach it. Next is protein preparation wizard. In this we will find out the protein, and under this you will see that there is a import and processes, a review and modify, and in the last there is a refinement. In the first condition, in the first, that is your import and processes. First step is to import the protein. How the protein is imported? Protein is imported by the PDB, that is your PDB ID, protein which will come from the protein data bank. Firstly you select the protein, what kind of protein you are buying, like COX protein, PI3K protein, and GLP protein, PPAR proteins. What are the proteins you are going to discover or to find out? Under this we'll find out these. There is a 4-word code under the protein data bank. This is a site, that is protein data bank. If in the Google you will just find out the protein data bank, and you will find out the various proteins under these. Okay, then there is a 4-word code, then you will add this in the PDB ID and import the proteins. Firstly you will find out the protein structure. You will just add to the protein ID and going to the pre-process. Then your pre-proteins or the protein ID, after the adding the protein ID or you import the import protein, it will structurally like this. Then there are different protein preparation wizards. In the first wizard we're going to after that the pre-process. In the second in the first process we will import and pre-process, and what we will done in this process, we will just hit the pre-process button, and then it will automatically pre-process your protein receptors. Then in the next step there is a review and a refinement, and then there is a review and modify. In this you will modify your proteins. They are both in this protein, there are following chains: one chain A, chain B, chain C. There is a different kind of proteins or chains are present in which receptor. Where the receptor is present, then you will find out or you select those chain, and remaining chains will be deleted. Then in the last and refined process, you will just hit the optimize button and just click on the optimize button, and your protein will be optimized and seen like this. This protein is after the importation, and after that we are going to the sitemap. Sitemap is a process, or it is a channel, or it is a you can say a protein, it is an application of the search engine software. In this we will find out where our protein ligand, our protein will bind with the receptors. Like if you are going to see that there are different kind of ligands are attached with the receptors. Okay, under the sitemap condition you just find out the best docking pose or best receptor site where our ligand will going to the bind it. Next, there are the followings are the sitemap process. In the sitemap, the ligand will attach or ligand find out the best way or best pose in the receptors. The next is receptor grid generation. Firstly, you will find out the receptor. What kind of receptor? Firstly, you will find out the receptor in the ligand molecule or in the protein molecule. Then you just hit the receptor molecule, and there is a grid generation. From the purple color, you just find out or you see in the screen there are the purple color grid. Then it means that your drug or the drug molecules or the ligand molecules will go and repeat and will attach with these ligands in which your atom is selected. Next is the docking process. Under the docking process, we are going to hit the dock, and by the refining, in the docking process we'll find out our ligand is going to attach with what? Its receptor. It will go and find that the receptor is attached with the ligand, or we can easily or say the drug is bind with its receptors. Then under the molecular docking examples, in the last we'll study that, or we'll find that there's a following example. We'll going to there are following examples, that the drug 1, drug 2, drug 3, drug 4, drug 5 and drug 6. These are the examples of the different compounds. If we'll go to the drug 1, we'll find out the dog score. In the last I will earlier explain that what is the dogs in the dog under the dog score. It will the best docking pose. The more negative the dog score or glide score, the most binding pose or the most reliable pose will find out by the your ligand molecule with its receptor. As we can say that the drug 1 has the various negative, that is 6.83 of the score, and the docking score is under calculated by the kilocalorie per mole. Now these are the 3D interactions of the docking poses. In this, our ligand molecule is attached with its receptor. Receptor comes from its protein. Now I will explain following molecules in the PP 1 to 1. First molecule is PP 1 to 1 and its interaction between the PI3K molecule protein. That is the PDP ID comes by the 1E7B, as I told you under the protein preparation wizard, and this 1E7B is taken by the protein data bank. And after that docking, we will find out the docking score is the minus 6.83 kilocalorie per mole. And what is the another one? The ABP1PH Cl3 interact with the PI3K molecule, and the earlier I said that the PDB ID which is comes from the protein data bank, that is 1E7B. The docking, what is the docking score? If we are talking about the docking score, then this is the minus 6.64 kilocalorie per mole. So with this, I am stop my words here, and thank you.